Hi students, in today's class we are going to learn the chapter 2.3 which is testing the factors which are essential for photosynthesis. Okay, here we are going to conduct some experiments and here we are going to mainly conduct some experiments. Okay, so before testing the factors that are essential for photosynthesis, first we need to know what are the factors. What are the factors essential for photosynthesis? So to easily identify where it comes from, we can see that in our last class we have learned that the easy equation like for photosynthesis, 6 molecules of CO2 plus 6 molecules of water in front of sunlight and chlorophyll, it will form glucose C6H12O6 and 6 molecules of oxygen, right? This is the equation. So from this equation we can find some factors essential for photosynthesis. So this is the process of photosynthesis, right? So here these are the factors that are essential. So CO2, H2O, sunlight, and chlorophyll. 1, 2, 3, 4. And about these 4 factors, we are going to test whether these 4 factors are needed or not for photosynthesis. Okay. So now we know that water, CO2, sunlight, and chlorophyll are essential. That we know it just theoretically, right? So we don't know, is it sure? How do you know, is it essential or not? Why are you saying without water can't the plant do photosynthesis? We may ask, we may just think without sunlight can't the plant do photosynthesis. Likewise, we can just think. So now we are going to test whether is it needed or not. Okay, here this is the chapter here, and in first step, what is the basic and the first step is to do is keep the plant in a dark room for 48 hours or 2 days. Okay, 48 hours or 2 days. So we may ask, why should I do this? In the first step, why should I do this step? Why do I need to keep a plant, just a good nice plant which is blooming from our garden, which is a healthy plant? Why should I keep in the dark room? Likewise, so here here what the procedure of doing these experiments here. I'll just show that in common of the 3 experiments there's a common procedure. Okay, the first thing is we are going to check mainly this factor sunlight, then carbon dioxide, and also the thing is chlorophyll, right? And these factors will do a controlled experiment. We'll do for each, to check each factor, we'll do separately a controlled experiment. Okay, controlled experiment. You may be just say, how about this controlled experiment? Have you ever heard about that? Here in a controlled experiment there will be 2 setups. Okay, 2 setups will be there, and we will give all the factors equal for the 2 systems, and both system will be provided equal factors in equal amount, except the factor we are going to test on. Except the factor we are going to test. We are testing. Okay, so suppose if we are going to do a test to make sure that CO2 is essential for photosynthesis or not, we'll give all other factors in an equal manner. We'll give equal amount of sunlight for both. There's no bias. They all be that both systems will be treated equally. Okay, here this both system will be treated in other factors equally, and we'll give same amount of chlorophyll for both leaves. Okay, same amount of chlorophyll. Here also same amount of chlorophyll, and we'll give water will be given in the same amount for both. Okay, water will be given in the same amount for both system. Then we'll give CO2 for the factor we are going to check is CO2, right? So in a controlled experiment, the factor we are going to check will be given to one system and will not be given to the other system. So here we'll give CO2 for this system, and this system will not be provided by CO2. Here the CO2 will be absent. And after, likewise, we'll do for each and every experiment a controlled experiment. Okay, likewise, I have just shown how we'll do it for CO2. Likewise, we'll do it for sunlight and chlorophyll and everything. And there, after doing the experiment, we'll find out, we'll do a starch test for both, in both system. Okay, in both we'll do starch test. Here we'll do starch test, and here we'll do starch test, and finally we'll find out does this starch test answers or not. Here we'll if there is a color change for iodine and the starch test has been answered, and if there is no color change and the starch test has not been answered, that means there is no starch has formed. That means there is no photosynthesis took place. That is how we'll come to a conclusion, and this is the step taken for for all three experiments here. And the first step will be doing the most first step will be the most first step is will be keeping the plant in a dark room for 48 hours. That will be the most basic thing, most basic and the first thing will be happen we need to do. And the last step here, the last step will be a starch test, and from this result of starch test, the result will be after doing a, here we'll do a controlled experiment. Here we'll do a controlled experiment. Here we'll do a controlled experiment. It may differ for each factor. It may be in one style for sunlight, it will be another style for CO2, and another style for chlorophyll. And this step will never change. Okay, this step will never change. We need a plant which kept under dark room for 48 hours, and the final step also will never change. And what the final step is to do a starch test, and from the answer of the starch test, if it is positive or negative, we'll come to a conclusion. This is how we'll do a this is how we do a experiment here. And here, a controlled experiment is more important to make sure that other factors didn't affect the result. Okay, here the first thing we need to know is why we need to keep this plant under dark room for 48 hours. That's the question raised, right? So here we know, now we know, finally we do a starch test. That is for sure we'll do. And that's the result of starch test. Result of starch test will affect our conclusion, right? Result of starch test only affect our conclusion. That's for sure. So before doing the starch test, we must do more accurately this test, because this test is going to affect our result. That you must be more careful about this. And just think, you are do if you are taking a plant, a potted plant, okay, before um, before going into the experiment, why here we all use is a potted plant. Why we need a potted plant? Do you have thought of it? Here first we'll keep this plant in a dark room, okay. We'll keep in a dark room, and again we'll keep under sunlight. In each and every experiment we'll keep in a under sunlight again, okay. Then is it possible with a plant which is stick to the ground? Can we take it from the take it away from the sunlight and again keep it under the sunlight whenever we need? That's so hard. How can we do? Can't do that. We need to plug it and make it die, okay. We can't do another plant which is fixed to the ground, okay. We can't do this step for a plant which is stick to the ground. So we need a potted plant for sure. And the next thing is the final step is to do a starch test. And just imagine, uh, if you are just taking a plant leaf, you are taking a plant leaf, two plant leaves, two leaves from a same plant, not the two different leaves, two leaves from a same plant, and doing a starch test, okay. Starch test, and doing a starch test will give you a result. A positive result will be found, right? And the color change will be found. And that means they have got a star, they have got starch. We know already that the plant leaves will store the starch they have produced temporarily in leaves. And for now, if we take these leaves just like that and started to do an experiment, what will happen? Even if we do or not, if we do the experiment or not, the answer will be a positive for starch experiment, starch test, because they already have got some starch on them itself. So they will always show a positive result for the experiment. There is no difference. But so we don't know whether that we can't make sure whether that photosynthesis took place or not during the experiment. That is the important photosynthesis synthesis took place during the experiment. So if we do in these leaves, just like the way we just pluck from the leaf, just pluck from the plant, and if we do the experiment, they have already have some um, they already have some starch on them, so they'll always show a positive result for our experiment. So what we need to do is, we want to make sure if we do a test with just a leaf which is not kept under a dark room for 48 hours, we are just taking a plant which is kept in the light for day and day nights, and it is not maintained in the dark room, and we are just plucking the leaf. And if we do this experiment for two leaves, the result will be same. And if you say that experiment has affected, no, that the factor will affect the result, the people will not believe you. The people not accept you, because there is no proof that there is no starch found before the experiment. They may say that if you do the experiment with just leaves which is not kept under 40 dark room which for 48 hours, you just do the experiment, and you are doing, for example, you are doing for sunlight, and no sunlight, I'll just take CO2. And if you take for CO2 and you'll finish the experiment for one system, you'll give CO2, you'll you don't give CO2 for another system, you give light, equal amount of water and everything for both system. And now you have found, you'll do this experiment, that starch test for both, and you will find out that starch, both leaves will show that they have got starch, and the color change will be prominent. So but then you'll what is the conclusion you'll come for? Then you'll say, yeah, that CO2, the presence of CO2 will not affect photosynthesis. What CO2 is not essential for photosynthesis, like that you will come to a conclusion. So yeah, because sometimes CO2 is present or sometimes CO2 is not present, but in both there is starch is present, so you will come to this conclusion. But the truth is not that, because the starch which you have tested is that starch present before you stop giving the CO2, before the controlled experiment, is the starch that is found in the leaf has given this result. So what you must do is first create a starch free leaf, like sugar free, starch free leaf, starch free leaf. First you must make a starch free leaf. So to make a starch free leaf, we keep this plant under a dark room for 48 hours. This is the reason we keep the plant under a dark room for 48 hours, and make sure that plants that leaves the that starch found in leaves, stored in leaves, have removed. That starch found in leaves, that starch stored in leaves are removed, and after that we'll conduct the controlled experiment. So first, the first step, you must, in each and every experiment, the final step is to do a starch test. So you must make sure that you started the test with a starch free leaf and find starch in one leaf, or two or both or whatever. So first, to create a starch free leaf, you must keep the plant under a dark room for 48 hours or 2 days. This is the reason. So first we'll just note down the basic, and the first step is to keep the plant under in a dark room for 48 hours. Then the reason for why is to make a starch free leaf. It is easy to just make you this term starch free because you can remember easily, but this term is not mentioned in your book, so you can't use this word. You may say like to remove the starch stored in the leaf. So to just remember easily, to just keep in your mind like something interesting, and you just keep in mind like something to make starch free. Okay, and the next step, it will be different for each experiment. The next step is to do is about controlled experiment, right? And it is like different for to testing CO2, sunlight and chlorophyll. And The final step will be also always same. The final step is doing a starch test. Starch test. And these both steps are always same for three both experiments. And here all you need to see a different is within this step. Okay, and here they have omitted this step, and just they have noted like things in your things needed they have mentioned a potted plant kept in a dark room for 48 hours. Dark room for 48 hours. So they have, instead of this step, they have mentioned this. Okay. So you just keep those things in your mind so you can easily note whenever you ask. Okay. Now we are going to do the experiment for sunlight. This is the first one. So for sunlight, we have given the things we needed are as usual. What a potted plant kept in the dark for 48 hours, and we all knew that always the final step is to do a starch test. So the things needed to perform a starch test, they have mentioned simply. So you may know what are the things that needed for starch test. You know, alcohol, water bath, and Bunsen burner, iodine, and everything. You can just keep it on your mind. Okay. So whenever if you are given like a things needed for starch test or given like separately, you can just make it clear. Okay. And the thing here we are going to study is about sunlight, right? Sunlight. Sunlight. So here the thing for sunlight, the special thing for sunlight here they are given is some special things needed are uh, a black and a colorless polythene. Okay, black and a colorless polythene. This is the thing you need here. So now we are going to perform the experiment, and here you'll take the plant which is kept under the dark room for 48 hours. Just show you how. Here is the plant. Okay. So okay. Now you wanted to choose two leaves which you are going to perform the experiment. So here we need to choose uh, almost similar leaves. Okay, almost similar, because we know that other factors must be given in equal amount for each plant. So water must be given in an equal amount for both setup, and both chlorophyll wanted to be in equal amount for both setup. So we can't do that those things accurately. Wherever green leaves are available, the chlorophyll will be available, and water will be given when the plant is alive, right? The root will get the water for the plant. So we can choose two leaves which are basically in a same position, like a with the same area, with the same size. Likewise, we can choose two leaves. So I'll choose these two leaves here. I'll choose these two leaves here. Okay. Now we are going to look at these two leaves. And now what, what we need to do is to easily to identify the leaves, I will just name those leaves with letters or numbers or whatever. So I'll name with leaf number 1 and leaf number 2. Okay. Now the black color polythene and colorless polythene are given for you. So you'll keep one. You'll stick black color polythene for one leaf. I now here I have stick that black color polythene for leaf 1. Okay, I have not for the whole leaf, for just a part. Okay, and wherever you want you can stick the black polythene in that leaf, and in the other polythene you'll you'll cover a certain part with the colorless polythene. Okay, now I have done the setup. Now I need to take this plant under the sunlight to let the plant to do photosynthesis, because we without sunlight we need to, in a controlled experiment means we need to give sunlight for one system, right? So we'll give the sunlight, let the plant do photosynthesis. Okay, now I have taken the plant under sunlight. Yeah. So these are the leaves, right? And this, and here I have covered them, right? This one is colorless one, and leaf 2. Leaf 1 will be black colored one. So now we'll let the plant to do photosynthesis. So we need to leave the plant for 3 to 5 hours under the sunlight, because we need to give a certain time to perform photosynthesis, right? We can't we can't give just 4 minutes or 5 minutes or 30 minutes. It's not enough. So we must make sure that photosynthesis have took place, right? So we just give the plant about 3 to 5 hours, just an average time for this plant. And if you don't let the plant enough time and just give just 4 or 5 minutes, or just like 30 minutes, and if you take the plant back and do the experiment, the starch test, and the result, the both plant will show the result. What's the result the post plant will show is uh, both plant will show the negative answer for starch test, and there will be no color change for starch test, and the negative answer will be found, and the starch test will be not answered if you don't give enough time, because there will be no starch will be formed, because there is, you didn't give enough time to produce in food. Okay, so there will be no starch will be produced. If there is no starch, how will you get a positive answer for starch test? It is can't. So there you will don't get a positive result in any leaf. So now you must leave enough time without hurrying. You must leave 3 to 5 hours to let the plant do photosynthesis. And after finishing the 3 to 5 hours, you will take the plant back to the lab. Okay, lab, and you'll cut off the leaves and do starch test. Okay, yeah, you leaf. This one is leaf one, leaf two. This is the part which was covered by black polythene. You noticed that what you have done by covering the plant with a black polythene? Okay, I'll say. Here you have given enough sunlight for this leaf, and this is a colorless polythene. So this here the sunlight can go through this, because this is colorless and it is transparent. So the sunlight can go through this. So here you have provided sunlight. Here you have provided sunlight. Paste the other things. Now I'll show. Here you have given those two leaves are here, and this is how you have given both of them. And here you have put a black polythene, and here you have put a colorless polythene. So here you have given enough sunlight for this leaf. It means here you have given you have covered with a colorless polythene, right? Colorless polythene. So the sunlight can go through this. This colorless one means it is transparent. So here sunlight can go through this, and in this leaf enough sunlight has been given. Okay, here sunlight is present. For this leaf, sunlight is given. And what about this portion? Have you here you have hide this part with a black polythene, so sunlight can't go through this. Sunlight can't go through this. Sunlight will not go through this part. It will go through this part. It will go through this part, but this part will not be not will get sunlight. So here you have stopped sunlight for this leaves as a particular part of this leaf. So here you have made that controlled experiment. Okay, so now I wanted to do a starch test back, and here I'll remove the polythene and um, I'll put enough iodine for both leaves. Okay, we'll put iodine for both leaves and just take a look about the result, and we'll notice that this leaf, that leaf number 2 will show color change. Starch test. Starch. Okay, starch. So color change. Well, if you think about color change, this leaf will uh, in this leaf a certain part which was covered by black polythene will not show a color change, and other parts will show a color change. And in this leaf, the whole leaf will show color change. Okay, so if this leaf, the whole leaf, if this whole leaf show the color change, that means here starch was present. Okay, starch is present here, and that means through this colorless polythene that sunlight has been given, and the starch has the photosynthesis has took place. And what about this leaf will show like, this leaf will be like this leaf. Okay, and this leaf will be fully in dark blue, and color change is prominent. And here the other parts which was not covered by the black polythene will show a color change. And what about the part covered by a polythene, black polythene, that didn't get any sunlight, therefore there will be no color change. Okay, so you can make sure that here that sunlight is important for photosynthesis. If you stop providing sunlight, so you can't, that starch will be not produced, and that means photosynthesis will not take place. So here comes your conclusion, and you will now you have tested the first factor, is it important or not. Okay, you have tested that sunlight is important or not for photosynthesis. Now we need to do other two experiments, uh, which is to test whether carbon dioxide is important, and other one is chlorophyll and everything. And we'll do that experiment, we'll continue about that experiment in our next class, and I'll stop this with with this one experiment, and it will be continued in next video.